All right. Good morning, everyone. I am Kailing, the Regional Field Application Scientist of Azure Biosystems. Today, we are having the second session of Azure Cielo Real-Time PCR system in 2021. I'll be mentioning the word Azure Cielo a lot of time today, but today we will be focusing on 3 application nodes and also the analysis module used in the analysis software. Right, first, let me introduce Azure Biosystems to you. We are a United States-based company in the Silicon Valley, or near the Silicon Valley at the United States, California. We are a team of highly experienced scientists, engineers, and entrepreneurs dedicated to accelerate your science. By innovating in unexpected places, we create products that combine smart, simple workflows with high performance and affordability. The result is that you can have the confidence in your data to move forward quickly and the flexible capabilities to go everywhere that the data takes you. So in short, we aim to give you the raw data that you can rely on using a simple workflows that you can trust. Right, this is a flow chart of what have been happened over the years. Our company founded since 2013. Right at the moment, we have 3 key important instruments: the Sapphire biomolecular imager, Azure imaging system that both of them are focusing on protein expression imaging, and this is our star today, Azure Cielo Real-Time PCR system, which we just launched last year. This is our agenda for the session. Like I said, we will talk about 3 application nodes. Firstly, about 6-channel multiplexing, sensitive and accurate single copy amplicon detection, and the third one will be how Cielo eliminates the need for passive reference die. And next, I will show you how the Cielo device software looks like and also the analysis module in the analysis software. In short, this is how Azure Cielo looks like. It is an open platform supporting both dye and probe-based chemistry in Qpcr reaction. In terms of performance, Azure Cielo has a unique optical design that delivers more sensitivity in reproducible data. It is flexible. It is engineered for a wide variety of Qpcr application with high sensitivity. And in short term, it is compatible with a broad range of dyes. Our user interface on this standalone machine is a very simple touchscreen access setup to make your run very easy and reproducible. And this machine does not require external PC to control it to run. But you do need external PC to analyze the data though. Okay, in short, this machine can transfer the data by using internet connected via Ethernet, LAN cable, Wi-Fi, or you can just transfer the data via USB port. So this is how it looks like in a Cielo machine. We have this called innovative and high performance optical design. It's designed around individual well scanning or individual total well scanning. It comprises 2 sets of 16 fiber optics. One set, 16 of them, are excitation fibers coming from the high-powered channel-specific LED to the excitation light source, to excite the dyes. Then you have another set of 16 emission fiber going to the camera, the detector camera, and also accompanied with the filter lens. So these 2 times 8, so 2 times 8 fiber optic module, so ensure that the specific wells on the Qpcr plates are evenly illuminate, like you can see over here on the left, at the same time, therefore increasing sensitivity and reducing the background noises. We have 2 configurations. We have 3 and 6 channels, 2 different models. which are compatible with a wide spectrum of Qpcr dye base. The machines are calibrated to all these dyes, so they are optimized for it in terms of performance and also sensitivity and background noises. And in case you have custom dyes and probes, as long as they are compatible with the excitation and emission wavelength, they can be easily calibrated into the machine using an intuitive one-step procedure. In short, Cielo is capable to give you interval uniformity based on and also give you the temperature accuracy, ensure that your Qpcr run is consistent from time to time. It can detect up to 2-fold differences in gene expression, and also multiplex without or low signal crosstalk, and it has melt curve analysis, reproducible for run-to-run up to 1,000 continuous run, and also a broad application compatibility, which fits into these 3 biggest criteria of a good Qpcr system, which are giving you a good performance, trustworthy data, and also the flexibility to do a wide variety of gene expression application. Right, let us move into the first application node, which is 6-channel multiplex Qpcr using Azure Cielo Real-Time PCR system. Let us start with a very little bit of introduction. Multiplex Qpcr, as you can see here, I have a figure showing you how the multiplex Qpcr looks like. Basically, you have a single well, or that you normally assign with single sample, and you can detect up to, in Cielo, you can detect up to 6 targets per time. Each of them have different probe. So this multiplex Qpcr offers a few advantages for investigators and scientists to study more than just one target of interest. You can do up to 2, 3, 4, and 6. This will save you time and money. It provides the results more quickly, and it also minimizes the consumable consumption because you don't need to put so much, use so much plates and consumable Qpcr mix anymore. And you can obtain more information per sample. This is exceptionally useful if you have a very scarce sample which is hard to obtain, but you want to get the most data out of it. And these RNA levels and gene copy numbers are compared directly within the same reaction. So you don't need another controls. Let's say I'm doing gene A in this plate and gene B in another plate. You do not need a lot of control to normalize and control these 2 different reactions at different plate at different time. Does not require controls to account for the pipetting differences between the wells or plates. So these are the advantages for multiplex Qpcr can offer. It uses target-specific probes with different fluorophores labels on the multiplex Qpcr. First, you do not need to have this the Qpcr product to have different size. So it eliminates the needs for Qpcr product to be differently sized. And there's also no interference from non-specific signals that can arise from the secondary PCR products binding to the intercalating dye, such as cyber green. So you don't need to design additional consideration in terms of primer design, and then you do not worry about the signal interference. Right. How do multiplex Qpcr utilize in scientific worlds? One very case that is dear to us, which is the COVID-19 virus detection and differentiating between the influenza A and B virus. And it right now at the current stage, it can use up to 5 plex Qpcr, which is a lot, because you want to differentiate 3 viruses at the same time in the sample. And of course, multiplexing Qpcr also applied in gene expression study, rapid detection and differentiation of potential pathogen in a biological sample, just like the COVID-19 infection, and also optimized for use in microgravity on the International Space Station. Right, how we do the data, produce the data in this application node. So this is the method. So if we are performing 6-plex RT-qPCR reaction to simultaneously detect 6 mRNAs in a sample of human cDNA. And of course, this is, we need to generate standard curves. So each of them have the standard curve generated by performing a cDNA dilution series. The two-fold dilution series of a human reference cDNA are ranging from 250 nanogram up to down to 1.95 nanogram per reaction, and then all these dilution was assayed in a duplicate. On the right, you can see these are the targets, the 6 mRNA targets that we quantify in the same sample. In this dilution series, we have RRP36, beta-actin, RNase-P, GAPDH, Tana box binding protein, and also eukaryote elongation factor 1 alpha 1. These are mostly the controls that we use. Right. In that, we prepared a master mix solution in such that each contains 20 microliter of reaction volume, containing a final concentration of 1 times a multiplex supermix Qpcr mixture, and also 0.5 microliter of each of the 6 primer and probe assay mixtures. After you seal the plate, the PCR conducts according to this protocol. We have the initialization at 95 degrees Celsius for 1 minute and 30 seconds, denaturation step at 90 degrees Celsius for 15 seconds, annealing and extension step at 60 degrees Celsius and 20 seconds. So the step B and C, denaturing and annealing, will be repeated for 45 cycles. Then the data were extracted and analyzed using Azure Cielo Manager absolute quantification mode to identify the CQ values and generate standard curves for each of the 6 channels. So this is the data where we pull out the single individual amplification curve. This graph is called amplification curve. Multiplex Qpcr reaction was scanned for all these 6 channels in this method, in this application node, I would say methodology. And these are the single channel amplification curves. And you can see that there is no crosstalk or signal leakage between one another. The reason of this is because we have the filtered lens that calibrated and perfectly optimized for each of the dyes, thus it gives you such a no crosstalk multiplexing experience. Right, so this is a left, you have 6 standard curves, each detecting different target. Right, so it's a linear dynamic range covering from the 250 nanogram down to 1.95 nanogram total input mRNA. Across, this is actually 8 orders of magnitude. Each of the reactions showed a very excellent efficiency, as you can see on the right of the table here, nearly 100%. 100% means your Qpcr efficiency is very good, as expected and is supposed to be, has a 2-fold increment at every single cycle. All right, so this is how we count the Qbc efficiency based on the standard curves and how the target amplification, signal amplification per cycle. So the efficiencies are good. The R-square values are very close to 1 across the range of the target cDNA used. It shows that the Cielo is capable of giving you such an exceptional performance with high efficiencies and linearity over a wide dynamic range. These are the references I'm we use or mentioned in the application note. If you're interested about the International Space Station, how they use multiplex Qpcr, you can check out this publication for more details. Right, let's move to the second application note. Azure Cielo optical designs eliminate the need for passive reference time. Okay, we come here is a little bit of introduction. So when Qpcr were first introduced, many of them use a stationary light. On the right, you can see the figure, representative figures, telling you how it looks like in the imaging in the conventional real-time PCR system. Traditionally or conventionally, they use a stationary light source, one light source, to scan or to image the whole 96 well together simultaneously. Of course, it has its own advantages and also disadvantages. So what is the disadvantages here? Because you have a different optical path. So this is a detector, the camera detector on the side. Each of them have different optical path at each of the well because due to the location of the wells all across the 96 plate design. Thus, scientists started to use reference dyes such as rocks to normalize this varying optical path. This is in the conventional real-time PCR system. So how it affects in terms of the data? Variation in the absolute fluorescence measurement is highly dependent on where the well is located on the plate. For example, if you have a well such as here, which is in theory shorter than the other one, for example, this is A1, shorter than the well in the middle. The well in the middle will appear, or it will have higher fluorescence reading than this well on the edge, regardless of the actual fluorescence signals at that point in the cycle. This is quite, I would say, not, this is, I would say, the biggest disadvantages of this type of optical design, because it does not really represent what is a fluorescence signals anymore, because it still boils down to the optical path where you put your well or your sample. It matters. So thus we use rocks, also known as carbonyl is a widely used inert fluorescence dye that is commonly added to a Qpcr reaction mixture to normalize these fluorescence signals, because in theory each of them should have the same amount of rocks, and rocks is not affected by Qpcr reaction. It remains stable throughout the entire Qpcr process, and provide the attributes of the Qpcr mixture are kept constant. So you have everything nicely done. The only thing they can change it is only the pipetting skill. If your pipetting skill is bad, of course your rocks number will be high or low, right? So we're not talking about human factors here. I'm just talking about rocks. So rocks is good. It is not affected by the Qpcr reaction. It's very inert, but it also has a superior spectral properties that can be used as one of the fluorescence dye in Qpcr reaction. Thus, rocks itself could have been used to detect a target of interest. Why this is very important nowadays? Because with applications such as the detection of influenza A and B and the COVID-19 G target, you can see this detection kit, and these SARS-CoV-2 and influenza A/B are the Qbc detection kits. They are using 5-plex dye reaction to differentiate and detect 3 of these viruses. This is in high demand and is more and more common right now, thus highlights the need for an alternative solution, because if we can change something in terms of the optical design that allows us to use rocks as one of the dye channel, would it be good? And you have one more option in terms of multiplexing. Okay, so here's the method in how we produce the data. We have 10 replicates at a 20 microliter of final reaction volume per well. Each of them has 5 microliter of 2 times human genomic DNA, 1 microliter of human GAPDH primer pairs, 10 microliter of no rocks Qpcr mixture, and also 4 microliter of Qpcr grade water. So where the human genomic DNA comes from is diluted from 0.2 milligram per mil diluted into 10 nanogram per microliter to serve as the nucleic acid template. After you prepare the master mix, aliquot them into 10 different wells, seal the plate, conduct the Qpcr according to this protocol. All right, I will not go into details. Okay, now let us go into the result. So the result, you can see that all 10 of them has a very tight and uniform amplification curve. So this is an amplification curve. So it shows that the Cielo system can uniformly detect amplification at every Qpcr wells. The real-time amplification curves of all 10 replicate wells shows a uniform increase of signal detected and giving a CQ mean value of 17.54 with a standard deviation as low as 0.02. That is very low from the statistical analysis. And the coefficient of variance, CV, of these CQ values is 0.13%. Like I said last week, what it means in terms of CV values is that acceptable range for Qpcr is 30%, 20 to 30%, 30%, I'll say. 20 to 30% is good, it's okay. 10 to 20% is very good. Below 10% is exceptionally good in terms of Qpcr data. So Cielo can give you 10 replicates as low, the CV values as low as 0.13. It is very super good, all right? It reflects the excellent precision of the data and how it's able to achieve such a uniformity in terms of the signal detection. It has two different factors here. First, we have the optical system, the fiber optic, scanning the whole single well here, and it obtains up to 100,000 pixels per well. And of course, we should not neglect the uniformity in terms of heating temperature. Temperature accuracy is also one of the factors that gives you that uniformity data. Right. Using rocks in a Qpcr reaction is very helpful. It has been very useful for the past few years in correcting the signal variation in instruments that utilize a stationary light source and also a stationary detector camera, and also the whole plate imaging system. What in the Cielo is we have 16 well fiber optic system, 2 sets. One set's coming from the excitation, another set's going to the detector's camera. It minimizes the variation within the 10 replicates well. Thus, we eliminate the previously commonplace signal variability caused by the optical path variation. That's the reason why we say Cielo can able to give you the precise and reproducible Qpcr every time without the use of extra dies. Right, these are the references used in this application note. Right, we have talked about 6-channel multiplexing Qpcr. The uniform detection eliminates the needs for passive reference die. This is what we're talking about, is to eliminate the optical path variation. Next, let us push the limit of this system. Look at the sensitive and accurate single copy amplicon detection in the Azure Cielo Real-Time PCR system. Okay, so in this figure, you can see we have 2 sets of 16 fiber optics. One set is excitation fibers coming from the high-powered channel specific LED, and another set is that 16 fiber emission fibers going to the detection, which is a CMOS camera, and it captures approximately 100,000 pixels per data or data points per well. So what it gives is because it's scanning the whole well, individual well, and scanning the whole of it. It gives you more accurate data, more data points, and also lower noise because the light doesn't shine out the well at all. This individual well detection is better than a single photo detector system that shine on the whole plate and capture the whole plate image, which basically record one measurement per well. Right. One very important thing about Qpcr, especially when you want to reliably detect very low target copy numbers, is assay efficiency. Assay efficiency is one of the factors, and then there are different minor factors that determine your assay efficiency. Firstly, of course, your primer specificity. It must be specific, does not give you unspecific bindings that disrupt the whole Qpcr reaction. And second, you need to have a prop specificity, so you know that the prop is specific binding to your amplicons. It does not bind anywhere else. And finally, the determining factor for assay efficiency, one of them is the instrument sensitivity to detect when the prop signal increases above the background noise. Ideally, we do not want a system that could not detect the differences in signals, and the signal will be drowned by the background noises. We normally do not want that because it's very hard to get the data, and it's very hard to get the data and also analyze it and get a sensible conclusion out of it. Right, being all other reaction components being equal, here I say about the pipetting skills, operator, and also primer specificity is good, your prop specificity is good, and everything else are good and equal. Higher sensitive instrument will give you earlier detection, or you can translate into earlier CQ values. What it means is it shows you that you need fewer Qpcr cycles to get the signals that you can see and start to make data out of it. Right. These are the method. This is the method we use, the protocol. So we prepare the master mix containing the PCR components and one copy of the target DNA sequence per 20 microlith reaction. We prepare master mix and we aliquot them identically 20 microliths of them into all 96 well and in a 96 well plate. And then this is the list of final composition of each of the reaction. So we have the primer mix, primer set, and also your templates DNA. Right. Then you seal the PCR plate and carry out the protocol accordingly based on the manual. These are PCR cycle. We cycle it up to 50 cycle because this is a single copy DNA target. It's very hard to get a result out of it. Then duplicate plate will run together. So we have one running on Cielo, and then we have another one running on another Qpcr system, which I cannot disclose over here. Right. Then the data were collected and analyzed together using the software provided by each instrument. Of course, we set the algorithm the same, so we do not have any bias and differences in terms of analysis algorithm to determine the CQ values of each of the wells. Here's the data. You can see on the left, we have the table showing you that single copy amplification was detected in 88 wells, which is 92% of the plate running on Cielo machine, and 77 wells, which is 80% on the plate running on another system. So with a single copy reaction, there is a probability that you will not see the target, or the target will not make into some wells due to pipetting error and some other factors. So we are trying to push the limit here. What is the percentage rate for you to detect single copy amplification? Then we plotted a graph or bar chart over here, and can see that this is a CQ value plot graph. And we have a CQ value lower, means it's earlier. We have average 2 cycles earlier than the other machine, which is on Cielo will be 36.32, and the other machine is 38.53. And additionally, the standard deviation for the CQ values in Cielo is much smaller than the other machine. Representing Cielo gives you reproducibility among the replicates, even though you are targeting single copy detection, gene detection. The reason for this is also because of the optical design. Like I said, you have uniformity in terms of signal detection and also heat temperature accuracy. In addition, you can see comparing here, we have A coming from Cielo and B coming from the other machine. A showing you A is having earlier CQ value over here. The increase of the fluorescence seen in the other machine is actually later. We are going to pair it together. You see 20 and 20 here. Later in the other machine. So what it means is Cielo itself, because of the individual well scanning, it capture more data points. Thus, you have earlier CQ value. The signals are easier to capture and then translate into a data. Of course, you can get the data, increase the probability of detecting a single copy amplification. Right. Qpcr is known to become less reliable with decreasing target amount. We are pushing, in terms of Qpcr, the limitation can be up to 2-fold differences or 1.5-fold differences, but it is definitely more sensitive than regular PCR, which we use gel electrophoresis by the end of the PCR reaction to see the final product. Qpcr is definitely more sensitive, but if you say it is the most sensitive in gene expression, no, we have other wide variety of gene expression tools for you to determine the one single copy target, right? So this is very common sense in Qpcr community, which is we know that with decreasing the target amount with up to single copy and beacon detection, the Qpcr is becoming less reliable. But at the same time, we have a lot of modern application, even research and assay will push the limit of Qpcr, push the limit of this technology and demanding the higher performance. Thus, we have to figure out to use a good optical design and a good system to get the most sensible data out of it. These are the references we use in these particular application notes. Right, here we go back to the agenda. We have talked about 6-channel multiplex Qpcr, and without giving crosstalk in Cielo machine, Cielo can give you sensitive and accurate single copy amplicon detection. It eliminates the need for passive reference dye because of the optical design. We have 2 sets of fiber optics, which gives you a very good data transmission in terms of transmission and signal detection as well. Next, we move into, I'm going to show you how it looks like on the Cielo device software. What it means is the software used on this machine and also the analysis software that we use on a PC. Because this is a screenshot we have already shown last week, but let me go through it again. Cielo itself is can be a standalone machine. It's a compact system with an integrated touchscreen and protocol-based software. And you can define your run parameters and choose a channel to scan, reaction volume, lead temperature, and also an email feature if you want to receive the data, and also the notification of your run is completed to your email mailbox by the end of the reaction. And you can also set up your temperature and time with a very easy touch screen function. You can just touch the button and then edit the temperature and edit the time. And also the setup for melt curve is also on the same page, so it's very convenient. As the Qpcr progresses, you can monitor your Qpcr cycling with the current temperature, lead temperature, and sample temperature, and the completion time as well. And do not forget one of the perks of real-time PCR or Qpcr, which you can see the changes of the signal in every single cycle. So you can easily examine them on the machine itself, and also checking the temperature where monitoring the system, make sure that your experimental conditions do not change over the course of the whole Qpcr reaction. So this is quite easy and basic on the device itself. Now, after you finish your Qpcr run, when you get your data, we will move into the PC. PC is where you have the Cielo Manager software. It has a lot of key features listed over here. You can analyze, we have 3 different analysis mode at the moment, and compatible with probe and die base. And it has a very intuitive plate map design, so you can assign the information on the well. You can also control this standalone Cielo machine, setting up the protocols and everything, just as long as both of them connecting to the same internet or connected via the USB cable, right? And also, by that means, when you control it via the PC, you can view the real-time graph and the raw data, and also the temperature curve, and so on and so forth. Right. Next, we will move into the analysis part of the Cielo machine. In the Cielo machine, we have statistical analysis here, a very simple one, of course, and based on whatever information you assign on the well. So we have everything allows you to customize and export the report, and all the figures or graph in the Cielo Manager can be right-click and save or copy as an image file. We have a wide range of image file extensions such as BMP, JPEG, TIFF, and so on. And finally, if you want to use a third-party software to analyze the data, you can always do so. Just export the data in the raw fluorescence signal Excel file. And also, if you are doing your Qpcr according to my key guidelines, then we have the option for you to export it at different formats according to the RDML format, right? So this is a very brief looks at the Cielo software. Like I said here, the Cielo machine itself, you can receive email notification that containing the data itself, and also telling you that, oh, your run is completed on your email address once you set it up, so you do not need to use a USB and go plug the Cielo's machine. This, right? We have 3 analysis modules in Cielo Manager, Azure Cielo Manager software. Firstly, absolute quantification standard curve. I'll be showing this using screenshot because it's easier for me to explain. Right, so you can see here we have 3 types of analysis module: absolute quantification standard curve, relative quantification standard curve, and also relative quantification delta CT. Now let's look into the first one, absolute quantification standard curve. This is the data you will get on the left. This is amplification curve. Each of them are assigned, the colors are assigned based on the targets. You can see I assigned it Gene 1, Gene 2, Gene 3, Gene 4. There's amplification curve. We have the option to change it. This is a linear curve. You have the option to change it into log view as well. And the color can be set according to target or sample. Right, so you have the play layout on your right, you have your amplification curve on your left. In terms of analysis, we have 2 different algorithm modes to analyze your CQ value. By default, Cielo Manager uses regression mode to calculate your CQ value. Then the second option is threshold. Threshold is useful if you, let's say, using some of the kit or the manual required you to key in certain threshold value to determine or to eliminate the background noises in that particular kit. Of course, the kits are normally optimized based on the system, the platform, the Qpcr system itself. So they might have different threshold for different system. Right. As you can see, I mentioned again, we have two different modes for CQ algorithm, calculating the CQ value, regression, and threshold. So threshold, you can't have auto threshold, which will supposedly set the threshold value used to calculate the regression in the regression mode, calculate the CQ value in regression mode. You can also have the option to manually key in the threshold value, or you can drag and drop the line, the threshold value line here, to see the CQ value changes. Right, after you set up, click yes, then here you go, you get your threshold line on the left on the amplification curve. And you can also use log view to help you determine where's the background noises. And on the right, you can see there's an analysis data tab. We have three different types of data presented here. Firstly is a list. List is very easy. You can see we have the well name, the well sample name, target, fluorescence channel. What is the function of these? Are these standard curve, standard samples, or unknown sample? CQ value, CQ means CQ standard deviation, quantities. This is useful if you're doing standard curve quantification, and the melting temperature, and all these can be exported to Excel and any other file type. Right. On the left, let us go to standard curve tab. You can see that these are the standard curve that you will use in standard quantifications using standard curve. We have the standard curve plotted according to the CQ value detected in the target as different sizes. So they will plot against the log starting quantity and the CQ value. Then you get your efficiency and R square, the slope and y-intercept to allow you to calculate the unknown sample. So the data, when you set unknown in the analysis data, it will auto calculate the quantity based on the standard curve. All right, so yeah, all right, so here we go. We have the analysis data in 2D view. I'll show you how it looks like in Gene 1. So this is what you call heat map. Look, the redder it looks, it means your CQ value is higher. The greener it becomes, that means the CQ value is lesser. So it's easier and it's fast for you to check if your CQ values are working fine. For let's say this is what we know as negative control, or this is a positive control. Then you say, oh, so the CQ values are okay. So this is a quick check for you. Right. So the same data, the graph, 2D graph, heat map, we can translate into 3D format. So this is very good if you're doing presentation, and you can just right click and save it as image file. Same thing for the amplification curve, same thing for this heat map. Right. We have done about standard quantification, standard curve mode. Next, let us go to relative quantification, standard curve. There's a way to do relative quantification and also using standard curve at the same time. As you can see, this is a second mode. All right, so on the right, you can see the plate layout. Here we have two sets of samples, unknown sample, sample 1 and sample 2. Both of them are having one CMYC and also gap DH as an internal control. And you have another set that is standard curve, plotting the standard curve for each of them, CMYC and standard gap DH. On the left, you can see this is a very good amplification curve for, I think this is a 10-fold dilution series. Right, so here's a heat map where you can toggle between the gene or the target gene that you point, and then you check the CQ values accordingly. Right, so this is a quick check. Oh, okay, these are good, these are good. They are good having the very near and linear, very uniform CQ values. And on the left, this is how it looks like in the gene expression tab, where you can see the comparison between the sample 1 and sample 2. And sample 1 is the normalized reference sample. So you can see that the CMIC expression level is lesser in sample 2 when compared to the control. And on the right, we have the gene expression data. I have the target, the sample name here. CQ means normalized quantity. This is based on the standard curve and also the normalized quantity standard deviation. RQ minimum. This is the relative quantification value to show you the relative expression between one group and another. Yeah, here's the standard curve for CMIX, and we also have CapDH. All right, so this is pretty much for relative quantification, but also using standard curve method. Next, let us go to something we are very familiar in research and gene expression study, which is relative quantification delta CT. It's a third mode over here, and you can see we have a sample from wild type, and let's say this is a Qpcr cell line, and then I'm trying to quantify relative expression between of the step 1 between these two groups by using PGK1 gene as a control or internal control or normalized control, let's say. Right, so let us go straight to the gene expression. As you can see over here, the expression status is listed here. So you can see that we have more expression in the wild type than in the Qpcr cell line. That's it. So these are not true because I just assign it based on my previous research experience. And on the right, you can see the same expression data, the relative expression data, and as you can see, the graph is become smaller because I can hold this and drag to move to make the graph bigger and smaller so you can see fit according to your screen size. Right. So showing you how to do that. And same thing, this graph you can right click, copy in your Microsoft Office, paint everything, and also save as multiple images extension. Right. Melting curve is also very important. We have the melt curve threshold as well for certain application needs, right? So uniform melt curve means that you have a very specific amplicon that is melt at a similar temperature, right? So in summary, Azure Cielo has innovative optics and technology for high performance of real-time PCR or Qpcr reactions. It is flexible because it is an open platform compatible for both probe and dye-based assays, sensitive by the design, the optic fiber designs, and also the cameras coming from the LED, high channel LED, high power LED, and going to the camera. Reproducibility from run to run. This is also because of optic design and also the mallow pelter heating block that gives you the heating uniformity. And if the software is relatively simple, it's very easy for you to get the CQ values. These are the list of our selected customer that is currently in use using Cielo machine. We have Perkin-Elmer and others labs, and we have research institute all over the world in the US, in Taiwan, and also in Singapore, Kermin, which is doing food industry, Sariyoti in Indonesia doing food industry and cell, and other therapeutic or immunotherapeutic companies. Right, so we have customer reviews. You reviewing this machine on Select Science. Select Science is a very good platform. If you want to see how people rate their instruments over the years, we can go and check it out. And then what are the good and perks, and also what are the irks if certain instruments. And you can see here for Cielo, we have 2 different reviewers here telling us that Cielo able to solve their difficulties in Qpcr essays and application. Right, so this is a brochure of a Cielo. As I say, we have 2 different models. Comes with Cielo 3 comes with 3 channels, Cielo 6 comes with 6 channels. Calibrator for all these dies, so you can just plug and use. And then can be operating as a standalone machine, and is durable for continuous 1000 run and tested for power trip and everything, and CE certified. It can be connected via internet using Wi-Fi, Ethernet LAN cable, and also USB cable, so you can export the data. And also you can export your data using email, so which is very useful if you do not want to touch your Qpcr machine in a certain lab. All right, so we have the thermal performance using monopelter base block with 12 temperature gradient zones, and chemistry support open chemistry between 10 to 150 microliters, and sensitivity is up to 2-fold differences. On the right here are the 3 application nodes I mentioned just now: 6-channel multiplex Qpcr, sensitive and accurate single copy ambient detection, and also why the optical design is eliminating the need for passive reference site, because the optic fiber itself to solve the varying optical path in conventional Qpcr system. Right. Thank you so much. I will wrap up here the session here today. Any question about Qpcr or about Qpcr system and models, instrument, please feel free to contact us. You can contact us on here. We have our email address. I'm Kai Ling, the Regional Field Application Scientist. We have Nicole here with us today. Okay. And also Priscilla. So you can contact Priscilla with any question about the product and also application note. Yeah, so if you want to get the application note, you can also email Priscilla so she can send you the application notes. You can check out how we perform the data and collect the data and get the do the experiment. Okay, thank you everyone. And